Okay. Hi, everybody. Welcome to week 2 of spring term for Drexel AI. This is our second general body meeting. It's April 7th, 2021. And today we have 2 presentations, one from me talking about protein structure and a Python module I wrote in the summer last year to manipulate and encode proteins and how we're going to use that for our project. And we also have a presentation from Jeff about generalized adversarial, not adversarial.
It's generative adversarial networks.
Okay, there we go. Generative adversarial networks. So I think,
twister.
Yeah, it is. Jeff is going to go first to talk about the background and how we can use them to tackle this problem.
You want to go around and introduce people first?
Sure. So is there anybody who wasn't here last week? Speak up.
That's
We just want to hear. Okay. Would you like to share your major and interests?
Yeah, so my name is Arin Nguyen, and I'm computer science major at Drexel, and I'm a third-year student right now. I'm on co-op right now too.
Nice to meet you. You're doing co-op with DOS?
Yeah, with Dr. Das
Okay.
and another part-time co-op as like machine learning internship. Yeah.
Cool. I knew your name looked familiar. And then.
Hi, hello everyone. My name is Naif. Thanks for having us. This is my second quarter master in artificial intelligence and machine learning. I have more than 6 years experience in software development, DevOps, and project management. That's it. Thank you.
Oh, yeah. Welcome.
Okay. I can share my screen.
Sure.
You guys can see this. Sorry. Now you can see.
Yeah.
All right. So talking about generative adversarial networks. So what does this mean? Okay. So generative refers to basically we're generating, we have some data and we want to generate some data that looks like it, that's new. That's the generative part. So we're learning a generative model, basically and then there's an adversarial part, which is, the second part of the name that refers to how we're feeding examples into this network that are supposed to trick it. So it's sort of like a game between two different players, and you'll see and then obviously we use a deep neural network. That's the network part. So this is the architecture. So you can see you see my mouse cursor? Okay. So these are the real data samples that we're going to use. And then we have, the sample here that's coming out of this pool of images. And then we're going into a discriminator. And this is differentiable. So usually this is going to be a matrix combined with some activation function. So either like a rectified linear unit, which just means we're basically cutting off the zeros, and that might be something we want. We can decide about exactly which activation function we want later. But anyway, so this discriminates between real. So it's supposed to say these are the real ones and these are going to be the fake ones, and this is going to output a loss. So the difference between the expected value versus the actual value is going to be our loss. And then you can see the generator. So we have these latent random variables. So we're basically going to take a bunch of noise, so randomized data, and then we're going to multiply some function to it, or pass it through some function rather. And then that function is supposed to generate things that look like what's in here. So in our example, I think we're going to generate some random noise, and then the stuff we're going to generate is going to be some fake protein with some structural motif that we're trying to identify. So ideally, we want to generate samples that are new. So they won't be, but they're also valid. So like they could be from the real world pool, but we learn such that they can't be discriminated between real and fake. So in terms of like computer vision, this is an example of like what they can do over time. So you can see like in 2014, it was a pretty bad. And then this looks like, you know, highly cursed. And then, you know, this one looks a little better. This one looks like a real person. Yeah, so a little bit about like the discriminator. So this is what's called a supervised learning algorithm, which means we're using data and we're trying to derive some meaning from it. And this contrasts to unsupervised, where we are generating data to use. That's at least how I understand it. So the purpose is to discriminate between real and fake data samples. For our project, you know, this is going to be real data samples are going to be existing data samples in the pool that we use with a given structural motif. And then we want to generate new data samples that are perhaps not in the existing data that are also valid protein structures that also have this structural motif. And then our loss function, which is what we're going to use to judge the performance of our model. We're going to be minimizing the error in discrimination. So we want to make sure things that are fake are classified as fake, and then things that are real are classified as real. And then obviously there's going to be a penalty for misclassification. Yeah, so in terms of the generator, this is an unsupervised learning method, which means we're going to generate, we're going to use some random noise that's like taken from a distribution like one of these. And then we're going to operate on that data with some given weight matrix. So if we have n data samples, then we're going to have like an n by d, where d is the dimension of our data. We're going to multiply that by like a d by d weight matrix. And that will give us something that is transformed into something that looks like a new protein with our given structural motif. And so like, in contrast with our discriminator, we're trying to maximize the error in the discriminator. So we're feeding the discriminator data that looks exactly, it's supposed to look exactly like something that you might find in the pool of data we already have. And we're going to use information that we gain from like our loss function to update this matrix here. Also, feel free to like interrupt me if you have any questions. So we can see like loss functions are like this. So for our discriminator, we're trying to maximize the difference between or the sum. It doesn't actually really matter, but the sum between the log of whatever we get for our discriminator on the real data samples, and then the log of 1 minus the, so this is like the discriminator operating on like the generated data sample given a latent representation C. Latent representation just refers to like something that could represent our new data sample. So obviously, or clearly, like the generator loss function looks very similar, except we're just trying to minimize instead of maximize. So our like put these two together, we get like a minimizing minimax. It's called a minimax loss function.
Could you explain? I know there's the log and then there's the 1 minus. Could you explain those two parts?
What exactly?
So what's the purpose of the log? Is that to?
Oh, so like if we have so our loss function is outputting. Sorry. Okay, so like this is going to output something that's like between a 0 and a 1. That's going to be our loss. Like we're going to classify it as real or fake based on the classification like that. So if we get a loss of like 0, or if this outputs a 0, then like that means that this is very close, and like we're gonna get a log of 1, which is just 0. So that's gonna.
Okay. And then is log just a scaling factor?
Yeah, I mean, log just makes it more convenient. This is actually something called like cross entropy loss, which is like a commonly used loss function. It's also related to like expectation maximization. So like you're maximizing or minimizing like the probability that something turns up in your data, in this case like a real data sample, or like yeah, I don't know if that answers your question, but
it does. Thank you.
Okay. Yeah, so okay, next is going to be like the real meat. Yeah, so this is taken from actually the paper that was written by Ian Goodfellow, who's like one of the fathers of deep learning, about generative adversarial networks. So we can see like we're gonna have a bunch of training iterations, which is basically like how many steps we want to update our gradient. So the gradient, sorry, I'll explain gradient descent just briefly. So we have a loss function, and then like we're going to take the gradient, which is essentially like the slope with respect to, say, our discriminator matrix. So like, and then because that's a maximization problem, we want to go in the direction that the slope is like increasing. So like the function's decreasing rather. So like maximizing this function is the same as like going along the geometric surface, you might say, until we hit a local maximum. There's also problems that you might like run into, like getting stuck at a local maximum. Say like there's one hill and then there's like a bigger hill right next to it. You don't want to get stuck at this hill. So you can use something called adaptive moment estimation, which is essentially giving you some momentum to get over that hump. But I can discuss that later. And then similarly, this is going to be like a gradient descent. So there's going to be like a little valley instead of a hill. We're using mini batch gradient descent, which means that we're going to evaluate like a set of M data samples. So this is like M noise data samples. So these are the ones that we're feeding into our generator. And then like M examples from our right. Yeah. So yeah. These are these are the real data samples. Sorry. And then like after we operate on M data samples, we're going to take what we've learned from the loss function and feed that back into our discriminator, and like learn stuff about like, oh, if we did this wrong, we want to adjust our function basically. So that's going to be like our weight matrix. And we're going to learn like, basically, we're going to ascend the stochastic gradient, meaning we're minimizing the error. Is this clear to everybody? I'm sorry.
I always think of gradient in the third dimension when you have two predictors and then you get another value, which would be the loss or objective function. And you're basically trying to, at the steepest point of a three-dimensional surface, you're going to increase in the direction that you're going to go in the direction at which the gradient is the steepest. So then you'll end up at either a max or a min, depending on whether it's ascent or descent. That's how I always think of it.
Right. Yeah. So like
And then,
the rule of updating this is basically we're going to add like part of this gradient every time step that we're learning. So like using something called a learning rate, which is basically a coefficient that tells you how much we're taking from a given batch and applying that to our gradient, if that makes sense. So like,
and then a mini batch is just is it some random sample of a batch?
yeah, I mean, like, so there's like, we can use batch gradient descent, which is like operating on all the data samples and then updating, but this is like dividing data samples into like, you know, a set of K subsets or something.
Okay, so instead of updating the gradients after n data samples, you update the gradient after K data
Yeah,
samples,
like
which and then K is in
like
M.
I think
K is
K
in N.
here is the number of divisions or something, right? Like between, so like if we have K different mini batches that are over the set of all of our samples, then we're going to do that for all of those, if that makes sense.
Hey,
that make sense?
Jeff, you have a question in the chat. I don't know if you want.
Oh, I'm sorry.
read it or
You can't see.
I know.
Yeah.
You can always speak up.
Before you answer that question, I was just gonna say regarding the mini batch. Yeah, the K times M should be just equal to the number of
Yeah, the number of data
samples that you have. Yeah.
Right, yeah.
So K is just the number of batches. I think it's better to understand the batched version versus non-batch version. And then you can just figure out the mini batch from there.
Sure, right, okay. So Naif says, regarding the use of GAN, we know that the purpose of it is to generate fake data set that are actually similar to the actual, so it just supports our model to learn. Oh, so you're asking like, generate, yeah.
I think he's asking if the GAN can be used for any other purpose.
Yeah.
Besides,
Aside from
generating fake data sets that actually similar to the actual one. I mean, I saw several projects that uses GAN for different reasons, but I don't think the results are reasonable. Or I'm just wondering if can we use the GAN for different purpose? For example, generating images that, for example, I don't know. I'm just curious that if
yeah,
we can use
yeah, I can yeah. So like basically what we do with our GAN and what we are learning in terms of like this game between generating fake data samples and discriminating between real and fake ones has a lot to do with how we structure our data. So we could have some matrix that we multiply by our data that tells us, it's supposed to like tell us like, you know, is this a valid sequence of amino acids for this protein? That might be something we want to consider, you know, or like, that actually is like, I think what our project is getting at. Like we want to generate fake data samples that have some additional constraints on like the sorts of things we can generate. Like we want to generate only valid protein structures. I'm not actually like well-versed in like protein structures like Ethan is. So that's part of what I want him to talk about in his presentation.
I think what Yit posted was a good link. I guess instead of inputting, Yit can explain, but I guess the idea is instead of inputting, there's some instead of inputting random noise, you are inputting something that's kind of like a diagram or a sketch of something related to an output, and then
Okay, yeah, that makes sense. Yeah, there's no reason you couldn't feed something other than noise into here, I guess, and then like, you know, the amount that your generator affects and like shapes that into something that looks like a sampler from here is you know, is always learned by training your weight matrix for G.
Mhm.
Yeah, so I talked about this as the discriminator gradient descent. So this is going to be the gradient that we're using to update our D matrix or function. You can think about it as a function. And then we're doing sort of the same thing with these Z. So using like multivariate or like vector calculus, we can derive these gradient rules by, you know, separating this into functions. We can take the gradient with respect to D, which is like, and then multiply that by the gradient of D with respect to G, and then, you know, and so on and so on. So part of the parameters we need to consider are like the batch size we want to use, like we talked about. So this M, this is sort of a high level stuff that we can do once we actually have something implemented. But learning rate affects the ability of your gradient descent algorithm to converge to like a value that makes sense. So like you don't want something above, I don't think usually above 1. So like, anyway, we can, this is kind of like beside the point, but this is like really important, like the features that we're representing. I'm still not clear on that, so I hope Ethan talks about those. And then, you know, activation functions, like we could use a tanh function to map our values between -1 and 1, or something like a sigmoid function, which maps our values between 0 and 1. All of those are useful for different things. So activation functions, by the way, are things you basically like can append to a weight matrix multiplication. So like after we multiply Z by this G, then we can add a second step here. And that'll like sort of shape our data so that it's better fitted to go into our discriminator and has the same range of values, say, as stuff that's coming out of here. Yeah, so, and then like techniques to consider, there's Atom, which I briefly talked about in regards to avoiding vanishing or exploding gradients. So this has to do with like choosing the right learning rate such that you're not adding so much every update that it like explodes into something that is meaningless, so that it's not going to converge to like 0, say, if it's a loss function, or it's not going to, it's going to go into like the stratosphere basically, or like cause an overflow on your computer even, is like a potential danger. And then one-hot encoding. So this is something that I thought about because we're, I'm sorry, something that, yeah, so like one-hot encoding can be used to represent like categorical data. So if we have a sequence of proteins, then we might want to represent, and like we have a finite set of proteins that we can have in our sequence at any given point. We might want to, you know, split that up into features that say like, oh, this is a feature that represents like if we have a given protein at this or we don't. So like that's going to be a 1 or a 0. And like that's sort of the one-hot encoding. So we would have like, you know, many features for the same like space in the sequence of in the protein.
I've used one-hot encoding for the atom type or the element, whether it's a carbon, nitrogen, oxygen. sulfur, phosphorus. I've also used it for the positional atom type, so where it's located in protein, in the amino acid, and I'll mention that.
Okay. Yeah, and then also like we can add, I think we can add some structure. I think this is sort of what he was getting at, but we don't have to feed just noise into our generator. We can feed stuff that is already like sort of valid, I guess. But we also might want to like talk about, we don't want our generator to generate things that don't make sense, basically, is what I'm saying. Even if they're like easy for the discriminator to, how do I say this? Like, even if the discriminator can make them, like, can recognize that they're fake, they shouldn't be like stuff that doesn't make any sense at all, is what I'm saying. And then also, maybe we could do something with like a deep convolutional GAN, which is an added operation. This is used a lot in like image data, but you can also, I think, like impose some structure on other types of data, like weather data is a common application, I think, or to like temporally, like time series data. It's also something that's been used for it, but we can talk about that if that comes up. Yeah, so we have a, here are some slides that I consulted, and then like here's the original paper. I'll upload this presentation to the Slack. And then, yeah, there's a mathy article on towards data science that I think would be helpful if anybody's interested. And then, yeah, so if nobody has any questions, then we can go on to Ethan.
Thank you very much, Jeff. Yeah, if anybody has any questions, just speak up. For a, first model of this term, it certainly isn't like the most naive or the most simple. So it's okay to have some questions.
Yeah, I have a question. So instead of generating new data, and this question is more for you because it's like about proteins, can we use like unlabeled protein sequences instead of generating new data?
When you mean unlabeled?
Okay, what I mean is like we don't know all of the existing proteins right now. Like maybe we know like 1% of existing proteins,
Mm-hmm.
And what I say is like we already have the labeled data and unlabeled data from a sample, and what like can we use these unlabeled data instead of generating new data?
So you're trying to say, okay, we have proteins that we know exist, right, that are valid.
Yeah.
And those are annotated, and you want to use a GAN to generate more valid proteins.
Yeah.
I think we could like train our generator to label data if that's what we wanted. Like if we had like a sufficiently large like set of unlabeled proteins, then that we could train our generator to do that. But like I think it's hard to train a generator to operate on different types of data. So we wouldn't want to, I might be wrong about that, but like we don't want like just random noise to be fed in and then also like, unlabeled protein sequences. So we'd have to choose between one or the other. That makes sense.
My power's back. It's great. Oh. It's phenomenal. My computer was going to die too, so we're a-okay. Yeah, so I think what Molly was talking about is in the scope of the project, because we want to generate. Sorry, I'm moving it down. We want to generate valid proteins, but we want these proteins to have some sort of functional identifier, whether it be a specific function or it be identified by like a structural motif. So maybe as a first step, we'd want to, be be generating valid proteins and then add in the functional aspect of it, right?
Yeah.
Because you have, if you imagine overall, we want to be generating valid proteins, but then a subset of that is we want to be generating valid proteins that have a specific function.
Sure.
I was also thinking that it'd be cool to explore whether we could give a GAN non-optimally folded proteins and see if it could predict optimally folded proteins. So instead of it being noise, it's a protein with a different conformation.
So we would be rearranging the structure, is that right?
You'd be moving the atoms. Yes.
I understand.
Yeah. And I can talk a bit about the idea behind optimally folded proteins and non-optimally folded proteins now. Okay, I think I'll start with this. Okay, so this is what a protein looks like. Rarely online, or really, you rarely see this representation of proteins. You typically only see amino acids, or you might see the RNA that codes for a given protein. So the amino acid sequence is given by a string of letters, and then those amino acids inside cells fold, and they fold into, and in this way, or at least we think they fold into this way. You have this primary structure of amino acids that are connected by polypeptide bonds, and then you have secondary structure, which are identified mostly by alpha helices and beta sheets. These are helices where you have hydrogen bonding occurring right here and right here. And then these beta plated sheets, when they're stacked against another, there's typically like hydrogen bonds like this way, if you can see my mouse cursor. And then there's a third type of structure for secondary. It's called a turn. So if you can imagine, you'd connect these two structures by having amino acids going this way, connecting the two, so that people sometimes classify them into those three groups. And then the tertiary structure is how those 2 or 3 secondary structures fold in on each other. So you can see that there might be an alpha helix here and a beta sheet there. And then a quaternary structure is just multiple tertiary structures coming together, right? So hemoglobin has 4 tertiary structures that combine into a quaternary structure, and each of the tertiary structures has a heme group, which is what brings oxygen around the body, and that's where the oxygen is carried. So going down into the amino acids, we have the structure of an amino acid. You have an amine group, a carboxyl group. Typically, you elongate and form polypeptide bonds on this side and this side. And the R group is basically a radical group, and that's what identifies the type of amino acid. So this carbon that's attached to the R group is called the alpha carbon, sometimes identified by a Ca. And then we have the beta carbon, which is identified by a Cb. And then any other carbons on the radical group are identified by Cc, CD, Ce, and so on in the data. And so here is a slur of amino acids, none of which you'll have to ever memorize, but you can see how they come in very different forms. You have polar side chains, you have positively charged ones, negatively charged ones, and hydrophobic. Hydrophobic means that it's water hating, so those amino acids tend to be facing inside the structure, whereas hydrophilic, water loving amino acids are facing outside the structure. So sometimes when you're looking at protein structure prediction, you can use where what hydrophobic and hydrophilic amino acids you have to kind of fold the protein yourself, but that's a very difficult task. So does anybody have any questions about protein structure?
I have a question.
Okay.
So radicals is free radicals, like stuff that doesn't have a electron.
All this R means that it's a placeholder for, when I say radical group, I mean what's connected to that carbon. So for instance, you have that carbon, the alpha carbon's right here on histidine, and then the radical group is just right here. And for instance, yeah, that's all it is. There isn't, it's not a free radical. It's just whatever's attached to this alpha carbon is the radical group. For instance, right here, if you're not familiar with the skeleton structure of molecules, I'm sorry, but there isn't anything branching off the alpha carbon right here, which indicates that there's actually two hydrogens there. So glycine doesn't have any carbons attached, doesn't have any carbons in its radical group, because it's just, there's a hydrogen right there. There's two hydrogens right there, actually. Yeah, what's the most important part is knowing the atoms that are relevant. So nitrogen, oxygen, carbon, and then the positional atom types, alpha carbon, beta carbon, and then any other carbon identified with the CC, CD, or CE. And we can use those labels, those positional atom types, as well as the positions of those atoms. So the geometric positions in 3D space to form predictions. So I will transition over to this project I did over the summer and talk a bit about what I did, the approach I had, and where I think we can go from this work. So, right, I showed you in this slide, this is a protein. Let's assume that it's a protein with a native structure. What do I mean by native structure? Basically, this protein folded inside the cell into the shape because this shape is the most stable. It's the most thermodynamically stable, which means that if you move any atom out of position for this protein structure at this instant, then you'll be increasing the potential. So this has the lowest potential at this state. You can kind of think it like that. And so when we have native structures, you can kind of imagine that they're at the bottom of a curve where the curve represents energy. And then you have decoy structures, right? That occur when you move or shift atoms out of position. So for instance, this decoy structure is farther into the red, so it's farther from the native structure. And then this decoy structure is closer to the native structure, because it's not so far into the red. So that's just kind of how I visualized it. And the idea with this project is to measure the distance from each decoy structure to its relative native structure. And that's identified by the distance along this imaginary energy curve. And then we can use different metrics to kind of define that, define this function, which measures this distance right here. And I used two convolution neural networks to do that, to predict similar metrics from two different representations of a protein. So the first representation of a protein is I basically took its positions in 3D space and encoded it into a 70 by 70 by 70 angstrom grid, where each of the objects in that 70 by 70 by 70 grid represents a 1 by 1 by 1 angstrom position in the protein structure. And then in each of those cubes, I mainly encoded three features. The first one was a one hot encoded vector for the generic atom type. And then the second one was one hot encoded vector for the positional atom type that I talked about earlier. And the second one was either an occupancy value or a logical value. A logical value is the easiest to understand. It's just whether or not there's an atom in that cube. The occupancy value identifies the distance to the nearest atom. So you can imagine that if you have a 1 by 1 by 1 angstrom cube, the atom might not be perfectly centered within that cube. So we use the radiuses of each of the atoms to figure out how much of that space is being occupied by that atom. The second representation is a simple 1D representation where I just used one hot encoded vectors for the amino acids. So you can kind of summarize what I did here by looking at a sample protein, a PTN, and we're encoding it either into that grid structure or a primary structure, so just the sequence of amino acids. And then you're either feeding it into a CNN, a 3D CNN, or a 1D CNN. The 3D CNN was predicting 3 metrics to measure the deviations in energy right here. So first I was measuring an energy calculated by Rosetta, which is a bioinformatics package that models 3-dimensional molecular structures. The second one was a protein contact map, which is basically an n by n matrix of pairwise distances between atoms in the protein. And then this one is the MSE of 2 contact maps, the contact map of a decoy structure relative to its native structure. And then simply for this one, I was going to predict the contact map. And so the main getaway from this project was that the losses of these models were able to converge in the training and validation sets. So from that, I was able to conclude that at very best, the models I built were able to learn these metrics. It's very difficult to compare this method to the conventional way of finding protein structure, because the conventional way of determining protein structure involves looking at basically doing energy minimization, which is the idea of moving points around until you decrease the energy of, you basically have a minimum energy, decreasing the position of the atoms until you have a minimum energy, which is very expensive, and I did not have enough time to do that. And this is an example of what this last one-dimensional prediction was able to do, where it took in the amino acid sequence and it was able to predict with fairly good accuracy. You can see the root mean squared distance here of 2 different proteins, and these are the best case and worst case scenario in a data set of 1,000 structures. You can see that this one is very good. This is I think less than, I'm trying to think. It's less. It's like, I know angstroms are smaller than nanometers, right? So it's 0.09 nanometers, right? And this one is still pretty good, but it's not as great as this one. And you kind of see it still retains the shape, but it's just farther off. So using what I built in Python for this project, we can kind of adapt the use of the methods and then the model we apply to use a GAN. And I still have the project GitHub. So that's what I was going to present on next. But does anybody have any questions about the general flow of what I was doing here? Sure.
Can you explain how you got RMST value again?
Yeah, so yeah, that's something I glossed over. So I was predicting the protein contact map, right? So you can actually rescale, you can use multi-dimensional scaling to reconstruct the 3-dimensional structure. And then I used linear algebra to align 2 structures. And then you can calculate the distance between each of the atoms. You do the root mean square distance between each of the atoms in 3D space. Jeff, did you have a question?
Oh yeah, you were talking about how like the other method of finding native structure is minimizing the energy. Didn't you mention that?
Mm-hmm.
So like, I guess I don't understand how the method you did is not doing that. Like you're talking about finding the native structure as like finding the folding that such that like if you move any part of the atom, it increases the amount of energy. So isn't that like minimizing the energy anyway?
Well, okay, so sorry if I wasn't clear. This doesn't find the native structure. It measures the distance from the native structure. So the idea was that you could use these metrics as a gradient to discover the native structure.
Oh, okay, I see. Sorry.
So if you have some space, some curve in space, right, and you get a high distance, you can kind of use that as a way to apply gradient descent to find the native structure. I just never got around to applying that, because then I'd have to do a whole comparison between what's commonly used,
Right.
which is just tackling a very big search space. And I didn't explain this ahead of time, but the reason why protein structure prediction is so difficult is because for an n-length amino acid sequence, you have really 4 to the n possible permutations of an amino acid sequence. But then how each of those sequences in the 4 to the n fold has infinitely many dimensions, right? Not really infinitely many. Actually, yeah, it does have infinitely many, but it's not as bad because there is like an upper bound to that, where you may have noticed that I used 10 amino acid long segments, and I did that because the longest amino acid is tryptophan, I think, that is 7 angstroms long. So you can imagine the worst case is that you have to restrain yourself to a 70 by 70 by 70 grid if each amino acid is tryptophan stretched in a line. So that's how you would go about finding the upper bound to this. And then there were some optimization things I did, like getting rid of empty space around the atom, but still feeding it into the network. Any other questions? I was going to go into the Python module real quick, just to give an overview and show you some of the cool features that I was able to extract. If everyone's still interested. I know I am.
Ethan, how'd you get rid of the empty spaces?
Yeah, so it depends how the values were encoded. If it was a logical grid, then I just looked for the smallest prism that didn't cut any values, right? So I did like a search. You compress via the z-axis, compress via the y-axis, compress via the x-axis. And then I guess for occupancy, I just look for non-zero values. And then I did that for each of the samples, and then I just used the worst case, right? The one that is the largest of all the cut prisms. And then that greatly reduces the number of inputs you need to have onto the network, because you don't have to actually include whatever 70 cubed is.
What exactly is occupancy?
Yeah, so I found this in a paper, and it measures, I couldn't explain the mathematical function, but it's an exponential function that measures basically, okay, I'll read it. So for a given atom with a radius A and its respective van der Waals attractive force, it calculates kind of how much of that space is taken up by the nearest atom. Well, okay, this calculates the occupancy of a given atom. I found the nearest atom for every grid point, and then calculated the occupancy of that nearest atom. It's from a paper I grabbed in the summer, and I guess I read about and included it in the project. But
so it's not a volume occupancy, but energy occupancy? Because you're saying it's the van der Waals attractive force radius.
Yeah, so I guess there is a tie between energy occupancy, because it takes into account the attractive force radius of any given atom. So yeah, so there's a bit of a tie between the energy part, but it's not like you have to calculate this. It's not like you have to calculate the energy for the entire protein. You just do it for atoms at a time.
So are you interpolating like atoms onto the 70 by 70 grid that you're talking about? Like
Yeah.
the atoms, you find the occupancy value, and then like based on the closest point in this 3D space, you're like projecting like a representation onto that point. Is that what you're
Yeah.
saying?
Yeah. Because, right, what do you do with images? You use a matrix. What do you do for three-dimensional structures? You have to use a grid.
Do you encode like how like the difference between actual like, I guess like this 70 by 70 grid is supposed to like exemplify its position in 3D space with like the folding.
Yeah.
Right? So like, because you're working on a discrete grid like that, there's obviously going to be some inherent error in any representation that you use.
Yeah, that's why I tried to use this occupancy.
So, okay. So is the occupancy like telling you how different it is, like from like what it actually is? Is that?
Yeah, so if we're looking at like a carbon atom, I use the van der Waals attractive force, but then I use this distance, right? And if this distance is sufficiently large, then this goes to 0, right? I don't know if I did that right. I'd want this one to, so like I want this term to be 0, right? So if this term is large, then it'll go to 0, right?
Yeah. So if you're increasing the radius.
Yeah. I think so. Yeah. Most of the time I used the logical expression, so there was a lot of inherent error because I just assigned a 1 or 0 for whether or not the atom was within that space.
So there's not like, I guess what I'm trying to say is that like on the 70 by 70 by 70 angstrom grid, you're representing like what the protein structure that you found looks like. But like the native protein structure is not confined to like this discrete representation. How do you like rectify like, you know, small differences like in, I guess like, I don't understand how the occupancy value is representing like the distance between like something's nearest point and like its actual point. That's what I'm saying.
I mean, I could go into the code and figure out exactly what what I did.
Jeff, I don't think it represents I don't think, maybe I'm wrong, but occupancy, I think, does not represent the distance. It's just, I think, a proxy to the structure. And in order to get that proxy, you have to get two values, one from the distance between two atoms. So you get that the R sub A value. And R sub van der Waals is just a term that comes from energy, because I don't know the reason, but it seems like the energy is also another component that we need to take into consideration when we are looking at occupancy. So that means if you have 1/3 volume, so you have one atom space, second atom, you would think that the volume and occupancy would be same for, let's say, one carbon atom space, another carbon atom, but compared to one oxygen atom space, another oxygen atom. But apparently it is not, because not only you look at the distance between radius, but also you look at the attraction force between atoms. So obviously attraction between two oxygen will be different than the one between two carbon. So, and I think that makes sense because structure might be same for two protein. If I might be wrong in this, maybe you want to correct me, but for two proteins that have same structure but with different atoms, you should expect different functionality. So that's why I think we're also taking into the energy. With that being said, I think it would be very useful and helpful if you could just see what the data set looks like.
Yeah, I
Maybe
agree.
like in a CSV file, or the way that you represent grid in the code. If you have any way to write it to hard disk so that we could see it, that would be great. I think.
I was going to go through it if you guys would still like to see. So, all right, I'm using some things here. PTN is the protein structure. It's the protein object. And I was going to talk a bunch about how you can define kind of like the ID, and then you can define the start and the end. But all right, if we have start and I'm saying grab residue 9 up to residue 18, and by residue I just mean the amino acid. Make this larger. So then we can define the protein at that spot. And if the protein structure isn't downloaded from Protein Data Bank, it'll download it onto your local machine, and then we can access the identifier information like this. Oh yeah, we know that this is the information we gave to it. This is a 4-digit code corresponding to the structure. It's a unique identifier, which is interesting because it's only, I guess it makes sense. Whatever 4 to the 26th is. Sorry, 26 to the 4th. And you can always see where this is downloaded, and it just downloads a CIF file. I wasn't really concerned with this file representation because it really didn't help me. So I created functions that will give me, something like this. So this gives me all of the amino acids. And if we look at the first amino acid right here, right, I wanted at the very least, I wanted the positions of the carbon, alpha carbon, beta carbon, and nitrogen. The alpha carbon and beta carbon are the carbons that are not in the radical group, and then this carbon is the first carbon that appears in the radical group that I showed in here. So this is alpha carbon, this is beta carbon, and then this would be carbon right here. And then we were including the residue name, and then any other atoms that are in the structure, like the oxygen, or we have beta carbon again. And then what we can do is we can grab all of the coordinates of the protein structure, X, Y, Z, and then they perfectly correspond to the atoms that are represented in atom list right here. So CO, CB. And for those 9 residues or so, sorry, I meant to copy this. You see that there are 69 atoms. Okay. And we can filter by different atom types. So if we only want to use carbon atoms, or alpha carbon atoms, or beta carbon atoms, we can do that. And we can actually generate, we mostly use kind of like this, the coordinates. That's what kind of superimposes it onto the object structure. We can use that data to create metrics. So we might want to get a distance matrix, which is an n by n matrix of pairwise distances between each of the atoms. We could do that for all of the atoms, or all the alpha carbons, or all of the alpha and beta carbons. And then I wanted to show this. This calculates the pyrozeta energy. So it outputs a number which corresponds to an equation that pyrozeta has. So right now we calculate the pyrozeta energy be 17 and a few, and I can show you what it takes into account. So these are all the metrics that pyrozeta take into account. And this is why it's so difficult to do energy minimization, because a typical score function takes into account all of these different variables. It has like electron max distance, electron min distance, the diameter of the electrons, yeah, and a lot of other features that I just haven't explored. So what's really cool is we can do some encoding, right? So if I want to take all of the amino acids and then go from the protein structure to grid, I can go right here, and it'll create a matrix. And that is just an object structure, right? It's going to take a bit to load, and I was going to show you how it's 70 by 70 and how it's just a list. It's just a list of objects, but I think what's the most important part is what we have offered for each of the atom objects. If I grab one atom object, the first one, I can show you what we have. So all of these values will be undefined because I'm essentially grabbing the upper left-hand corner of the protein structure, right? So if I go to like atom here, it won't have anything. It'll be none. Chords. That is just the center of that object. If I grab another one and I go for chords, it'll be shifted by that amount. So it always initializes it. It always initializes the object to the center of that grid, and then the occupancy is kind of what I took into is kind of what I use to determine the shifts. So might make sense more to generate a logical matrix. So it's just a compilation of zeros and ones. You don't see the ones here, but I can send this into a grid. I can send this into a function that helps me visualize the structure. And this is what the protein looks like when you just use logical values. I use this function to validate my decoy function, and this decoy function generates decoy proteins or non-native proteins. Basically, I apply a shift, a small random shift on all of the positions within the protein structure. And then using this calculation, I can generate the mean squared error between both contact maps. So like how I generated the contact map, like how I generated the distance matrix or contact map here, I just take the mean squared error between each of those. So you can see that this one is, the decoy structure is slightly off from the native structure that we loaded in originally. And then in this module, there are functions that automate the creation of data. And they load them into files. They load them into object files and text files for export that are easily implemented in like the CNN. So it'd be really cool. Yeah, you were saying, Jeff?
Yeah, so in terms of like orienting the protein structures in space, is that something that's done like uniquely for each like native structure, or is that like some standardized algorithm?
So the way that these coordinates are obtained is through, I think, X-ray crystallography, and then they're obtained. You can have different models of the protein. So you can imagine that really these proteins are dynamic, and so they move. So snapshots are taken in time of their movement. And on the website, they did have different models of the protein, but I just simplified and took the first one that appeared. And then I would look at like the first chain, which is identified by A. So the A chain might be this section of the protein. And yeah, so the positions of the proteins aren't unique at all. And I think I had some function in here that would, before processing the data into the CNN, I would actually randomly rotate the protein. So similarly, if you were having a CNN to recognize whether a hand was up, down, left, right, you'd want, that's a bad example. If you want,
you're learning like representations based on like holistic representations, not just one perspective.
Yeah, so like if you were using the MINCE data set, you might want to rotate the characters, rotate the digits a little bit. That's the idea. So I use rotation. I apply the rotation matrix. That's something we were talking about last week, because it was saying we could have just did a parametric rotation, which would have been easier, but I didn't know how to do that. But yeah, so this is the module. It's on my GitHub. and I have the demo, I think here. I'm blind, obviously. Right here with all the function I was running, and then a requirements file. The one thing I have to say is that if you want to use it, you have to download PyroZeta, and to get PyroZeta, you need to, it's not offered through pip, so you need to get a license, and the licenses are all free, but you get like a username and password sent to your email address. But if you just don't want to deal with PyroZeta, you can just not use the PyroZeta functions, like the ones that are right here, or the one that calculates the energy. Yeah, but that's it for me. Do we have any questions?
I guess like I know you were talking about like different ways we could use the GAN to solve different problems. I guess like which one are you leaning towards here?
So I think it'd be really cool to discriminate function and generate proteins with a specific function. And the first step in that would be to create a data set of real proteins of a specific function. But the trick would be to generate meaningful proteins from the generator, right?
Right, yeah. So like I was talking about like imposing some additional structure on like the stuff that we generate. That would be part of that.
So what's interesting is that, you know, a simple ANN or CNN predicts the structure of a small protein relatively well. It's not terrible. So maybe if we can use some larger version of this model for the generator or for the basis of the generator, that might work pretty well. Then it's just a matter of discovering ones that fit a particular structure or have a particular function.
The idea is that we're generating more data that has valid structures with the same function.
Mm-hmm.
And that's like the end goal of the.
That
like,
is the end goal.
If we like wrote a paper, that would be what we were.
Yeah, that'd be the paper's on. Yeah, cool. Well, yeah, that's it for me. I'll be sticking around for if there's any questions. But thank you very much for listening this week. Next week, I think he had offered, I'll talk to him. He offered to do a kind of a workshop on GitHub, and that'll help with the management of this project. So I guess stay tuned for that.
So when are we likely to start? Like, what's a estimated start week? I'll say like we would
Probably
start.
probably at the end of next meeting.
Okay, cool.
He'll put the tasks on GitHub and then get everybody who wants to participate oriented on what we could work on in the coming week. I want to get this started as soon as possible.
Yeah, me too. All right, this sounds awesome.
Thank you.
Thank you so much. Appreciate it. Thank you.
Yeah, of course. Thanks for listening in.
Yeah, thanks a lot.
Of course.
Thanks, guys.
Yeah, of course. Thanks for the presentation, Jeff. I really appreciated it. You'll post it on the general channel?
Yeah, I can I'll like export a PDF or something.
Okay, cool.
All right. Yeah, I guess I'll see you.
Actually, could you do me a favor?
Okay, sure. Yeah.
Could you wait right here? I'm gonna switch back to my Wi-Fi and hopefully it won't fuck up everything. So
Okay.
I'm pretty sure if I just disconnect, it'll still stay recording.
Oh, okay. Yeah.
Maybe if I hit pause recording and you're still here. Okay, I'll try that.